Today, I'm going to be talking about some very recent work from my group, looking at principles that may be shared across neuromodulation modalities. These are my disclosures and sources of support, and these slides, as well as the references, can be found online. So the story really begins in 2008 when I got another great honor from Dr. Paulus when he invited me to speak at this meeting that was held then. I think it was the third meeting. 2008 was also the year that Barack Obama was first elected president in this country here. And I remember traveling to Germany with great excitement to attend that meeting. And this is a slide that from my talk back then, over a decade ago, where I introduced some very recent work from my group then, developing models of transcranial direct current stimulation that were based on high resolution MRIs of individuals. And that work itself built on a decade of prior work from other groups. And the emphasis at the time, and continues to this day with these models, is that you need to segment the tissue into different compartments that have very different conductivities. So for example, skull is especially resistive, while cerebral spinal fluid is especially conductive. And so generating these models, it is most important to properly separate out between these tissues, because how they impact current flow from the surface of the head to the brain will be very different. And so really the emphasis from, that has always been there, has been capturing the right level of precision in these models in order to have them produce accurate predictions. And then this work appeared shortly afterward in brain stimulation. So since 2008 until now, there's been a lot of work from my group and others on making these models work better, optimizing them for different applications. And I can give just a few highlights. One, from 2011, was switching from thinking about just modeling many different montages and seeing how current flow reaches a given brain region of interest, and instead starting from the brain region of interest and asking where should we put the electrodes to optimally deliver the current there. And we and other groups have been working on this problem. There has been continuous validation of these models since 2008. This is a very important one, I think, where intracranial electrodes were in people. These were people with epilepsy undergoing routine clinical monitoring, but stimulation could be applied at the same time. And we were able to directly validate the same predictions that were made in 2008. That first model that I presented in 2008 took Abhishek Datta, who was then a PhD student in my lab, 3 years, like literally 3 years of work before the first model solved. Now, thanks to the work of many groups that have worked to disseminate these modeling tools, you could do it on the same laptop you're using to watch this talk in about 3 minutes. Download the software, and with limited background, you can simulate any montage on any head you want using ROAST or SIBNIBS or other freely available software tools. So many things have happened, both from my lab and others, but I want to say that for all this activity, in a sense, for all this change, some things did not change. Right? Sometimes like the more things change, the more they stay the same. And let me give you an example of that with one recent paper. This was just published this year. And what we thought about the space between the brain and the skull, which was traditionally modeled as cerebral spinal fluid. However, I don't need to tell this audience that that's not what's between the brain and the skull. While there is some cerebral spinal fluid, most of that space is actually occupied by different forms of meninges, like the dura mater. And these are significantly more resistive than the CSF. And so it seemed a mistake to assume that all the space between the brain and the skull was this highly conductive fluid. But you can't actually model all these layers. It's computationally intractable. So the solution we came up with is to emulate them. So instead of modeling the space between the brain and the skull as cerebral spinal fluid, we modeled it as this mixture of other things. And the way we did that was very simple. Instead of assigning CSF conductivity to that compartment, which was 1.65 Siemens per meter, we assigned to that compartment, which we now call emulated meninges, 0.85 Siemens per meter. And this was And moving forward, this is how in my lab we now model the head. This is probably one of the most significant changes in our workflow in a decade. And so the point is to show how, not innovative we can be, right? Where we've stuck, and not to take away from all the work in my lab and other labs that have done to enhance the precision of these models. But again, to some extent, we're still following the same recipes that have been established for decades. So I'm going to present new ways of doing computational current flow models that we've been thinking a lot about. And the story really begins not with transcranial direct current stimulation, but with a different technique called electroconvulsive therapy that, again, would be something that is very familiar to this audience. ECT begins with positioning electrodes on the scalp. There's certain classical montages that are used. Right, lateral, bilateral, bifrontal. So this is non-invasive electrical stimulation. But that's really where the similarity with tDCS ends, because while tDCS uses 1 milliamp of sustained current, ECT uses almost an amp, hundreds of milliamps, and they're applied in these brief pulses, and the pulses are repeated, and the number of pulses is typically increased until a seizure or a clinically meaningful seizure is produced. Now, the device measures the resistance of the head during ECT, and this is called the dynamic impedance, and it's about 200 ohms. So, for example, the voltage being produced by the device would be 900 milliamps times 200 ohms. So the 200 ohms is the resistance of the head experienced by the device. Prior to ECT, there's always a setup procedure. And in that procedure, the resistance of the system is also being checked prior to the ECT. This is done with a very low level of current. There's no desire to produce a seizure at this point. So it's about a 1 milliamp, but at very high frequency. And during this process, the head resistance that is measured by the device is called the static impedance, and it is higher than the dynamic impedance. So if dynamic impedance ranges between 100 and 300 ohms, static impedance can range between 300 and 1,000 ohms. So the resistance of the head looks different depending on what kind of current you're applying to it. And this idea is a very, very old idea. In fact, people have known about it since the foundations, the first use of ECT, where people were using voltage-controlled stimulation. So it's a very old idea. But the question is why? Why did the head resistance suddenly change? And there's two ways to think. And so if you wanted to model it, we would need to make a decision. We'd either need to make two models, one model for 1 milliamp and one model for 900 milliamp, where the head resistance was different. Or we might make a single model, but that model would have to change. That model would have to adapt to the current being applied. You would need to look differently under 1 milliamp of stimulation versus 900 millistimulation stimulation. And so that's what I'm going to be presenting to you. But before that, we did take a little bit of a step back. We did some retrospective analysis of data from different ECT trials. This is just one of them. Here, ECT is being applied either in the RUL montage or the BL, bilateral montage. Brief pulses, or what's called ultra brief pulses, are being applied. Each one of these dots represents a patient who got ECT, and that patient received multiple sessions, which is why you see a bit of a variance there. But it's obvious by eye, it's resolved statistically that there is a correlation between increasing static impedance and increasing dynamic impedance. And that relationship is maintained across these different conditions. So subjects that have a higher static impedance will also present with a higher dynamic impedance. So the two are related, and we wanted to, and it's been a very long-standing question as to if they're related, why are they related? And also, why are some subjects presenting a static impedance of 1,000 ohms, while others are only presenting a static impedance of 200 ohms? And what does that mean as far as the efficacy of ECT, if anything? So this could be addressed with, we hope the computational models can provide insight on these problems, and our approach was therefore to develop a single model that could model both these dynamic and static conditions. And so this is a new modeling pipeline, an adaptive modeling pipeline, where the tissue conductivity adapts with the current being applied to it. So I'm going to walk you through this pipeline. As always, we start with high resolution anatomical images. Now, these in this case are coming from patients who received ECT. And so we have clinical data from them, including their clinical static impedance and their clinical dynamic impedance. So subject-specific MRI, subject-specific clinical static and dynamic impedance. And as before, we divide the tissue up into different compartments that are important to us because they have different conductivities. An important step we're taking here is we're dividing the scalp into two compartments: a superficial scalp and a deep scalp. And then skull, meninges, gray and white matter as well. And current is applied to electrodes placed on the head, just like it's applied during ECT. As with prior approaches, we use numerical approaches, FEM, to solve the problem. This requires generating these meshes. There are many of them. Just in the skin, in the superficial skin, there are almost 2 million elements. And now we predict how current flow flows through the head. Now we do this for the two different conditions: the 1 milliamp, the static impedance situation, and the 900 milliamp, the therapeutic dynamic impedance. So in both cases, we predict the current flow that flows through the head. What I'm showing you here actually is the current flow on the surface of the head, or the electric field on the surface of the head. You see that 900 milliamps produces higher electric fields than 1 milliamp does. This pattern depends on the anatomy and on the conductivity that is assigned to each tissue. But here's where there's an important next step. The current, the electric field that is generated on the skin, changes the skin conductivity. The more electric field there is in any part of the skin, the higher the skin conductivity. And this relationship is captured through a transfer function that we developed based on prior measurements of how skin responds to current. So the more electric field any given part of the skin has, the more conductive that element of the skin does, up to a particular maximum. And for each subject, we're setting a different maximum. And so that is what's illustrated here by the initialization. This maximum superficial skin conductivity is specific, is an assumption. We assign it to that individual, and we also assign a deep skin conductivity that is specific. So now what we do is we generate what you see here. This is a plot of skin conductivity. And you can see now that skin conductivity is not uniform. In the case of the 1 milliamp case, you can see that it's increased around the perimeter of the electrode. Well, in the 900 milliamp case, you can see that skin conductivity has broadly increased across the forehead. So the current has increased the skin conductivity. But if the skin conductivity has increased, that's going to impact the resulting current flow. So now we need to resolve the current flow through the skin, through the whole head, through the brain, with these new conductivities. And so we actually get a new prediction of current flow, a new prediction of electric field on the surface of the skin. But that new prediction now leads to updated skin conductivities, which then change the current field. And so this loop is repeated. And while in the body, we assume that it's done essentially almost instantly, computationally, this requires thousands of iterations on a dedicated supercomputer before we converge to a solution where we're saying, well, during either 1 milliamp static ECT or 900 milliamp dynamic ECT, this is what we've settled on. This is the resulting current flow patterns, and this is the resulting skin conductivities. And that model also generates a prediction of that subject's static and dynamic impedance. So in the 1 milliamp case, what is the overall resistance of the head? And in the 900 milliamp case, what is the overall resistance of the head? The model generates that prediction. Now we actually have that data from the very same subject. So we can compare the clinical, experimentally derived static and dynamic impedance with the one predicted by the model. If they match, we're done. If they don't match, what we do is we go back into the model and we adjust the deep scalp conductivity and or the maximum superficial scalp conductivity. So we adjust the skin properties and we rerun the model and we predict a new static and dynamic impedance. And once again, we ask, does the model predicted static and dynamic impedance match the clinical static and dynamic impedance? If it does, we're done. And so now what we have is we're left with a deep skin conductivity and a superficial skin conductivity that is specific to that person. This is that individual's skin properties that lead to the correct static and dynamic impedance. So we are assuming that every individual, in addition to having a different anatomy, has different skin properties. Their skin is different, and it's their differences in skin that help determine the static impedance that is measured, the dynamic impedance that is measured, and so the overall current flow prediction. So I want to emphasize these variables that are subject specific. And so this, in a nutshell, and this is the reference where we explain this for the first time in some detail. It's a model that changes. Current flows through the head. The current changes the conductivity, which changes the current until we settle on until we converge onto a particular solution. This is what the result looks like. So this is a particular subject who received ECT. You can see that they have a clinically derived static impedance before ECT. They have a clinically derived dynamic impedance during ECT. And the model of their head also predicts a static impedance and a dynamic impedance that is relatively matched to the clinical case. And the reason it's matched is because we adjusted the subject's deep skin properties and superficial skin properties to match it. So those are these parameters that you're seeing here that were adjusted to make these things fit. What do the results specifically look like? That's shown here. So first, for the static model situation or the 1 milliamp situation, you can see the electric field on the skin. And then you can see the associated skin conductivity, which in this case goes up to about 0.035 Siemens per meter. And you can look at the dynamic model. Now here, even though the color scale is the same, notice that the label is different. The electric field is actually going up to 4,500 volts per meter on the skin. And if you look at the skin conductivity, there's that much more diffuse and intense increase in conductivity that goes up to 0.4 Siemens per meter. Why 0.4 Siemens per meter? Because that is the maximum skin conductivity of this subject. And the model will then predict the resulting brain current field. And we can look at another subject who had a different head anatomy, a different clinical static impedance, a different clinical dynamic impedance. Once again, the model fits this subject's clinical data by adjusting skin properties. So this won't work if everyone has the same skin properties. The model predicts that people have different deep skin properties and different superficial skin properties in order for the model to match the predicted static and dynamic impedance with the clinical dynamic impedance. And there you see the resulting predicted skin electric fields and skin current flows. And you can see that in this case, the subject's maximum skin conductivity went up to 0.3 Siemens per meter, because that's as conductive as their skin could get. Okay, so we then did something to try to understand why these models were behaving the way they were behaving. And what we're able to do in computer simulations is we're able to take a head with all the anatomy, and we can rip the skin off one head and we can put it on another head. And we actually can do this with both the superficial skin and the deep skin. So we're starting with the full heads, and these heads have different skin types, right, that we just parameterized. And we're taking these different skin types and we're ripping them off and we're putting them on different heads. And this is something obviously you could only do in a computational model. And so we're essentially creating new heads with one subject's anatomy, but another subject's skin. And so you create a mixture of these synthetic heads. And this is what the results look like. And the question we wanted to answer, by the way, in doing this was, what's the relative importance of head shape versus skin properties? So if you take someone else's skin and put it on my head, what is the relative importance of my overall head shape to impedance and brain current flow versus these skin properties that are different across individuals? And this is what the models predicted. Now, in A is the clinical data. This is from the same clinical series that we generated these models from. So A is clinical data. This is bifrontal ECT. And you can see that these subjects, there's a weak correlation, the black dots, between static impedance and dynamic impedance. And next then in panel B, you're seeing the result of these swaps. So these are synthetic heads where different skulls, different heads are being plastered with different skin properties. And what you see is you get this distribution, right? There's maybe a very weak correlation between static and dynamic impedance, but just like in the clinical data, there's also a very wide degree of dispersion. And the question is, where does this dispersion come from? It seems hard to understand, but again, with the models, we can unpack this data. And so what I'm plotting here is I'm fixing anatomy. So we're looking at the anatomy of subject 21778 in B1 on its own, and we're plastering on different skin properties. So each one of these dots represents a different skin that has been plastered onto subject 21778. And now a pattern emerges. One thing we can see is that if we fix deep skin conductivity, but change superficial skin conductivity, we change dynamic impedance, but not static impedance. Those are those vertically oriented points. What that means is that this maximum superficial skin conductivity affects dynamic impedance, but not static impedance. And that generates a dispersion in properties, right? Because now we don't have now you can see why that in itself would limit the relationship between static and dynamic impedance. But what you're seeing here on the lines that connect the points is a situation where the head is fixed, the head anatomy is fixed, the superficial skin properties are fixed, but the deep skin properties are changing. And you can see in this case, both static and dynamic impedance increase together. So they co-vary. And so this is why in this way, the models can explain both why there is a correlation, first of all, why subjects are different because their skins are different, why there is a correlation, and why that correlation is mixed. And so we think that individual scalp properties, if you look at these plots, count much more than head anatomy. So more than your head shape, your skull thickness, it is your scalp resistivity that's really going to determine where you sit on this plot. And we can also explain where correlations come from and why those correlations are limited. We've done more. We can look back at historical data. So what's being plotted here first is data collected by Umlauf and colleagues and Maxwell and colleagues. This would be 70 years ago at this point. And what they did is they applied ECT to subjects, but they decreased the amount of current and or voltage that they were applying. And so you can see on the x-axis that the current and voltage is being decreased, and impedance, and this is dynamic impedance, is being recorded. And Umlauf and Maxwell reported that as they used less and less intensity for ECT, the dynamic impedance went up and up and up. And this is not typically how ECT is applied now. Typically, pulses are changed, but they did do these experiments, and we can reproduce these experiments in the model. Where in the model we just apply ECT with less and less current and voltage. And so those are the model are the colored plots that you're seeing. And you can also see the experimental data both as a group and as individuals. And what's interesting is that most of this data fits a power law. The fact that not all these lines align perfectly makes perfect sense to us. We're simulating pulses. They use sinusoidal stimulation. But the consistency of this relationship was very compelling to us and suggested that the models were making predictions that novel predictions that were properly parameterized. But more than that, we're very enthusiastic because we think that these models may be providing answers to questions that have literally been asked for decades, like why does one subject have a higher static impedance than another? What will that mean for their dynamic impedance? What does that mean for the amount of current that's being delivered to the brain? And I'll refer you to this paper for all these questions. And I need to emphasize that these adaptive models where the conductivity of the tissue changes with current flow, they don't act like the standard non-adaptive models. I mean, for one thing, if you look at this plot, right, is a static traditional model, if you change the applied current or the applied voltage, the impedance it shows is always exactly the same, right? If you apply the impedance of a model, a traditional model doesn't change with how much intensity of current you apply to it. The whole point is that it's fixed. So the kind of behavior that you're seeing here would only be possible in an adaptive model. But more than that, we're seeing things like how current is actually delivered to the brain is different in these adaptive models than in the fixed models. And so I'm going to leave it here, and I want to change gears a little bit, and I want to go back to tDCS. And I want to ask the question, well, great, we have these new adaptive models of electroconvulsive therapy. What does this have to do with anything with models of tDCS or tACS or TRNS or other low intensity approaches? And there's a troubling clue that we became aware of very early on in this work, which is that there's this weird anomaly of how we're modeling skin in traditional tDCS and tACS models. Let me show you that. So in here, you can see that in the static model of ECT, which is 1 milliamp, skin conductivity was going up to about 0.035 Siemens per meter. It wasn't going up very much at all. When we applied a milliamp of current, 900 milliamps, only then did skin conductivity increase to about 0.4 Siemens per meter. And that's also consistent with recordings in the literature where skin properties were measured. You need very high currents to increase skin conductivity this much. But if you go back to the way we've been modeling tDCS for over a decade, you'll see that the values that we, and not everyone, but we and other labs have been assigning to skin are very high. We're treating skin during tDCS like it's ECT at 900 milliamps. The skin conductivity is much higher than it should be. And by the way, much higher than it should be if you go back to the original literature that measured skin properties. So during tDCS and tACS, the way we've been modeling it for over a decade is we're modeling the skin with a fixed conductivity, but as if it's getting ECT with a much higher conductivity than skin should have. So that's the question. Why, when we model tDCS, are we applying a scalp conductivity that should be reserved for high intensity ECT? And the answer, I think, is because maybe during tDCS and tACS, the skin actually is already adapting, just like it is in ECT. Now, why is that if it's such low current? Well, it's low current, but it's sustained, right? A key feature of tDCS since the pioneering work of Paulus and Nietzsche and others is that it's applied for tens of minutes in order to produce durable and lasting changes. And it turns out that when you apply stimulation for that long, not for a 1 millisecond pulse, right, but for seconds and for minutes, skin properties change. And this is something that was sort of right in front of us all along. In this paper, we were applying tDCS and measuring the voltage needed produced by the device, and so the subject resistance. Now, subject resistance impedance is shown here in green. And you can see that over the course of a couple hundred seconds of tDCS, the impedance of the head drops a lot. And by the way, after we turn off stimulation, it stays low. And this shows an adaptive response. It shows that the head resistance is changing slowly during the course of tDCS. So the skin has adapted. And when we initially did this work, we were more interested in the voltage. So that's what's shown here in the orange trace and the black traces. We were interested in how much voltage you need during tDCS because that has important implications for device design. But there's this underlying phenomenon here, which clearly shows that the head impedance is adapting. So we think that tDCS has this adaptive skin response already caked into it. And I'll also say, I think this may have important mechanistic insights, but just to return to the big question, why do we model tDCS like it's ECT? Well, because I think the skin is adapting, and therefore I think that these pipelines that we have developed for ECT should be applied for low intensity transcranial stimulation like tDCS and tACS, because adaptive models don't behave like static models. They don't produce the same kind of brain current flow patterns or intensities. And so perhaps this is something that will keep us busy over the next decade. I want to change gears a little bit, and I want to talk a little bit about neuronal network oscillation, something we have heard a lot about already at this conference. And it's something we've also been thinking a long time about. Back in 2008, when I was invited to speak at an earlier version of this conference, I talked about oscillations, and I talked about how we're very interested in oscillations because neurons are sensitive, synapses are sensitive, but networks are a lot of neurons and a lot of synapses together, and we have data showing that networks are more sensitive than individual cells or individual cellular compartments. So networks are the most sensitive to electrical stimulation, which is especially important when you're doing low intensity electrical stimulation. And we show data how these oscillations respond. This is a spectrogram of gamma oscillations in a rat. We show data how oscillations respond to direct current stimulation of different polarities. Adaptation here, by the way, means the adaptation of the network, not of the skin conductivity. This is data from a rat brain slice. We also looked at different frequencies, and we're able to generate I think, a relatively nuanced understanding of how at least this model system responds to different forms of stimulation, different frequencies, different intensities, different biases. And so we had a publication where we developed a detailed neuronal computational model parameterized by detailed experimental recording and brain slice of how oscillations respond to stimulation, and others have been working on the same problem. I want to jump now to recent work where we looked at temporal interference stimulation, something, again, I think this audience has at least heard of. With temporal interference stimulation, the basic principle is that you apply, let's say you stimulate on two sides of the head, and you apply two high frequency signals, let's say 1 kilohertz and 1.005 kilohertz. So both very high, but a little bit different. And the principle is that these frequencies collide in the middle of the brain to create a unique electric field pattern that may be effective in stimulating deep structures without stimulating superficial structures. So what you're seeing here is a computational model of temporal interference stimulation. On the surface of the head, you produce, in the surface of the cortex, you produce an unmodulated electric field. So a fixed amplitude electric field, just very, very, very high frequency, this kilohertz frequency, but the amplitude is not changing. Deep in the brain, you have regions of the brain where the electric field is still kilohertz frequency, but its amplitude is waxing and waning and increasing and decreasing. And so you produce this amplitude modulated kilohertz field. And so the temporal interference stimulation, it's not that no current is produced in the superficial brain. It's that in the superficial brain, the kilohertz is unmodulated, and deep in the brain, it is modulated. And it's this difference that has to underpin the ability to selectively stimulate the tissue only deep in the brain while sparing the superficial cortex. So this is something we looked at. We went back to our hippocampal brain slice model, these gamma oscillations. So you're seeing here a spectrogram in the brain slice. These oscillations are about 30 hertz, and you can see it's a rather steady power until we turn on stimulation. Now, as we've known for a long time, if you apply low frequency stimulation, like 5 hertz, you don't need a lot of intensity to produce a lot of modulation. So that's what you're seeing there in panel 8.1, low frequency, low intensity, lots of modulation. Now we can also apply modulated kilohertz, that's in A2, or unmodulated kilohertz, fixed amplitude kilohertz. That's what you see in A3, and this being a slice, we can co-vary a lot of different parameters, intensity, modulation frequency, carrier frequency, and so on. But generally, this is what we found. So we found this in this rat hippocampal slice model of oscillations and in a computational neuronal network model of the same system. We found, as we predicted, high sensitivity to low frequency stimulation. High sensitivity means you don't need a lot of volts per meter. Now, amplitude modulated kilohertz fields, which is what you're seeing in A2, modulated the oscillations, but they made them, again, wax and wane. So we call this a dynamic modulation, right? It wasn't necessarily that the oscillations got bigger or smaller throughout the whole time. It's that their intensity got bigger and smaller as this AM electric field went up and down. In contrast, the unmodulated kilohertz stimulation just produced a tonic elevation in oscillation power. That's what you're seeing in A3. So this is what we call static modulation. So both unmodulated kilohertz stimulation and amplitude modulated kilohertz stimulation affected oscillations, but they affected them differently, with one producing static modulation and one producing dynamic modulation. Now we can take this data from brain slice, couple it into a neuronal network model, and then couple that into a overall current flow model, and you get the kind of results you see here. And again, I would refer you to this recently accepted paper for more details. But what we're plotting here is the modulation of oscillations in the brain. And we're distinguishing between dynamic modulation and static modulation. So let's look, for example, at the first column, C.1, where 83 milliamps is being applied at the surface of the head through each one of these pairs of electrodes. And we are assuming a particular, some certain features of the neurons. That's what's shown in the time constant. And we're also assuming certain features of the network oscillations themselves. In this case, that GABA B is present. And C1, the first column, is really the ideal situation where what you see is that you produce dynamic modulation deep in the brain. That means the oscillations are waxing and waning. And superficially, you are producing a modulation, but it's a static modulation. So the oscillations on the surface of the head are just tonically boosted. It's not that they're not affected. It's just that they're tonically boosted. But in a tonic way, will deepen the brain, they're modulated. Now, the ability for this, as we can consider in this computational model, how much current you need to do this, and also the ability to modulate deep structures versus superficial structures depends on the underlying structure of the networks and the neurons that underlie it. So it is possible to stimulate selectively deeply, but in the sense that you're producing selective dynamic modulation deeply, while producing static modulation superficially. And this is sort of what we almost anticipated when we looked at these models to begin with. And the fact that oscillations are especially sensitive to stimulation was not new, but there is something interesting that I learned here, and that is that oscillations aren't just important to determine sensitivity, how much current you need in the scalp. Do you need 1 milliamp, 10 milliamps, 100 milliamps in the scalp in order to produce modulation, so sensitivity. But they can also have important implications for selectivity. So which brain regions are oscillating and which are not. And also the nature of all those oscillations themselves, the structure of the oscillations can actually govern selectivity as well. And I think this is a rule that a lesson that can generalize across neuromodulation approaches. The last thing I want to talk about is something that I'm calling neurovascular modulation. And I've been thinking a lot about this idea. And so I want to present it briefly in the few minutes I have left. There's three things I would like to share about neurovascular modulation. One is it has very important implications for how we think about current flow modeling. And the last two, I think it has very interesting implications for the outcomes of stimulation. First, about there's really only two biophysical principles that sort of everything falls out from. The first one is that the brain is riddled with capillaries, and very surprisingly to me, the average distance between these capillaries is 50 microns, right? And the soma is 20 microns. So it's a spaghetti. It's a spaghetti mix of neuronal processes and these capillaries. They are everywhere. And the other thing you need to know is that the blood-brain barrier, which is the sheath around the blood vessels, the thing that separates the blood from the brain, is extremely resistive. It's extremely resistive sort of by biological design. But so we have a relatively conductive brain, a conductive blood, and a very, very exceptionally resistive blood-brain barrier. So these are the physical principles that everything else falls from. Now, it's already very well known that neuromodulation affects vascular function. What's the most obvious effect of tDCS? Skin reddening. Many people have imaged the effects of neuromodulation, TMS, tDCS, and so on, using functional imaging techniques that are inherently dependent on hemodynamic responses, fMRI being the classic one. So you do tDCS, you use fMRI to measure brain signals. Now, fMRI is actually a change in blood supply, but as normally the case with fMRI, it's almost like that's like an epiphenomenon, right? The assumption is that fMRI is imaging neuronal activity. You're just then getting to it through the secondary increase in blood supply. And so the idea behind neurovascular modulation is that the stimulation is directly stimulating the blood supply. It's directly activating the vasculature. And then the neurons follow, meaning the blood is the target of neuromodulation. And so I want to unpack that through these principles. Now, the first principle, which is this notion that the presence of blood supply changes everything about current flow modeling, is a bit of a technical point. But typically when we do modeling, we model these macroscopic tissue properties like all of gray matter, all of white matter, all of skull, and then we predict current flow through the brain. But when the current flow arrives in a given voxel of the brain, right, let's say the motor strip or somewhere else, that motor strip is riddled with these capillaries, and they're very, very resistive. And so as a result of that, instead of the current sort of flowing through in a uniform way through the motor cortex, it has to twist and turn around these blood vessels. And this is something we've modeled in this paper. The preprint is available, and it's in Impress. And so for people who think a lot about how electrical current activates neurons, the fact that it's not flowing through uniformly, but the fact that it's twisting and turning has very profound effects for how we think about how neurons will be activated. In fact, the more it twists and turns, the more neurons will be activated. So I'll leave that idea at that and then refer you to this paper. But then the other piece of it is, well, could the stimulation be stimulating the blood vessels themselves, right? Why are we assuming that they're not activated? Could some of this current, instead of flowing around the blood vessels, flow through them, in and out? And if it does that, what will that mean as far as vascular response? So this is the second piece of it, and something that we've been thinking a lot about. And it was a little bit difficult to get at because there's a general thinking that neurons are like the prime players, and that vascular function follows, right? This is through classical neurovascular coupling, right? Neurons go first, and then blood vessels follow, but it has to be the neurons first. So if we electrically stimulate and we see a vascular response, we always attribute it to the neurons. So in order to prove that the blood vessels, the blood supply can respond directly, we had to resort to different animal models. This is one of those models. This is a dish with just cultured endothelial cells. It's a blood-brain barrier model. There's no neurons. So if this thing responds to stimulation, it has to be the endothelial cells directly. There's no neurons. In this case, there's no glia either. And these kind of model systems are very well established. We're applying electrical stimulation to them. We can measure different things like transport through this blood-brain barrier model, transport of molecules, but also transport of water. Water movement through the blood-brain barrier is tightly regulated. And in this experimental system, they do this with this little bubble. So the more water transport you have, the more this little bubble moves. And I'll refer you to the paper for all the details. But what we found was when you apply direct current stimulation to a blood-brain barrier model, the bubble moves. And what the bubble, that's that bubble displacement. And what bubble displacement means is that there's an increase in water transport across the blood-brain barrier. We did a lot of analysis of this phenomenon, and it is well explained by something called electroosmosis. Electroosmosis is inevitable when direct current passes through a tight barrier, a tight membrane. Water is, with certain properties, water is dragged with it. The tighter the barrier, the more water is dragged. It's interesting. The tighter it is, the more water you get. And the blood-brain barrier is exceptionally tight. And so you end up dragging a lot of water. To make a long story short and refer you to some key publications on it, I believe that during tDCS, electroosmosis is produced in the body, specifically across the blood-brain barrier. Water is dragged, and when water is being dragged, this triggers a whole cascade of secondary responses in the blood-brain barrier. We've shown that certain genes activate, very specific the transport of very specific molecules is increased. We've shown this both in vitro and in vivo. In separate experiments, so this is tDCS boosting transport across the blood-brain barrier. In separate experiments, we've also shown that tDCS actually enhances diffusivity through the extracellular space. What this work and other work that we reference is telling us all together is that it's almost like tDCS flushes the brain. It boosts brain clearance in a way that I think no other technique can, or in a very particular way. And what I'm saying here is that this is a new therapeutic target for old indications because this audience knows very well that tDCS is already being trialed for a range of diseases, neurodegenerative diseases, brain injury, and even psychiatric disorders where there is an association of disrupted brain clearance. In fact, any disease, I think, which is linked to disrupted sleep, and sleep is associated with brain clearance. You could bring in the notion that maybe boosting brain clearance will help treat the disease. And so what I think I'm showing you here is we're not suggesting that we should be doing clinical trials any differently. We're suggesting that there may be a very compelling and unique underlying marker. And this is just my final slide, an additional idea that bridges these things together, the notion of using tDCS in acute neuroprotection. So not in the chronic stage after brain injury, not in neurorehabilitation, but on an emergency basis while the brain injury is undergoing in order to protect it. There's been a lot of very compelling animal work showing this, and now there are some very compelling clinical trials addressing this issue. So this is one of them, and I don't usually reference ongoing trials, but I think this is an incredibly interesting one to watch. These are patients who have had a brain injury, and they're getting high definition tDCS within tens of minutes after arriving at the hospital. So it's an emergency intervention with a hypothesis that you should both inhibit the damage region but also increase blood flow to the penumbrum in an attempt to rescue it. And this study will have very compelling clinical outcomes as well as imaging outcomes showing how blood supply is rescued to these surrounding regions. So that is my time. Again, I want to thank everyone for your attention. I sincerely want to thank the organizers and